Hey guys, happy What Wednesday. If you remember from last week, I told you that you would be tested this week. Well, not really. We're just going to talk about a particular test that we do here at the lab. So therefore, we will be asking the question, what is an ELISA? ELISA? ELISA. So what is an ELISA? It is an enzyme-linked immunosorbent assay. So we're going to have some pictures of what it looks like and a couple of videos of Sophia showing you how it's done.
An ELISA is a plate-based assay technique designed for detecting and quantifying substances such as peptides, proteins, antibodies, and hormones. I am collecting symptomatic plant tissue to put into these mesh bags to mix with buffers that will then be crushed up and ground up so that we can release the pathogens from inside the plant tissues and into these buffers for detection. Once the buffers have and the plant tissue has all been ground up, we can then put it into the wells, and the plate with all the wells comes already pre-coated, and it's got the antibodies on the bottom layer, and then when we add the plant tissue, it will then incubate overnight, and when it's incubating, the plant tissue will then the pathogen, if it's present in the sample, will then bind to the antibodies overnight. And the next morning, when we come back to work, we will take a solution that we have created and mix together and add that after washing the wells to get rid of the plant tissue. We will the pathogen will still stay attached to the antibodies, and then I will add the antigen. And so the antigen, if the pathogen is present, will bind to the pathogen that's already there in the wells. From there, it will incubate on the shaker for about 2 hours, and after that, we add the color indicator. So, and then it will incubate again on the shaker for another couple hours.
The wells and plate will then be placed into a plate reader, which will generate raw data, and which will then have to be quantitatively analyzed. Wow, that was a lot of great information on ELISAs. So just to let you know real quick, we actually use the ELISA-based testing method to test for 3 different types of pathogens, Xylella, Phytophthora, and Xanthomonas. So if you ever have any questions about ELISAs or those particular pathogens, then don't hesitate to let us know. And I hope you enjoyed all this great information, and I hope it provided you some insight into the science behind some of your reports. So I will see you next week. Bye.